Hello and welcome to Nikolai's genetics lessons, and today's video going to be unusual because I got a problem from one of my subscribers, but when I start to analyze this problem I found a lot of mistakes in this problem. But funny thing that when I change all the numbers to realistic one, I still got the same answer because all the changes that I made are proportional. And for example, if we divide 100 by 10, we are going to get 10, right? If we reduce both this number and this number 10 times, we are going to get 10 divided by 1, and still the answer would be 10. So from the mechanical point of view, just pure mathematical, both answers would be the same, but from the point of view of biology, this data and this data are very different. And for example, for me as genetist, this data may seem unrealistic, impossible. So I communicate with a subscriber and ask if a subscriber want me to change the problem for numbers to be realistic, or want me to comment why this problem has unrealistic numbers, and then solve this problem with realistic numbers. So the purpose of this video basically to teach you that in biology you cannot just solve problems mechanically or just using pure math. You have to analyze the data, because this is very important. So here is a problem. The error rate of bacterial polymerase is 10 in the power of -7 mistakes generation. The size of bacterial genome is 10 in the power of 8 base pairs. The size of average gene is 2000 base pairs. How many genes are mutated per generation? Let's start with this number. So what basically that means? I do understand that many of my subscribers choose biology because they don't like math, and sometimes I have to explain even the most obvious and easiest things. So those people who are good with math, please bear with me for a couple minutes. So this basically means that we have to multiply 10 by itself 10 times. So we have to multiply 10 by 10 by 10, so 8 times. 1, 2, 3, 4, 5, 6. 2 more times. So this is going to be the same as 10 raised 8. And if we multiply all these numbers, we are going to get 1 with 8 zeros. So this is going to be also equal to 1 with 8 zeros. And as you see, this is 100 million base pairs. And this is absolutely unrealistic number for bacterial genome size. Compare with the size of E. coli, which is 4,600,000 base pairs. So as you see, this number is 20 times bigger than average size of bacteria. And today I checked literature, and I have found that the biggest bacterial genome size known is 13 million base pairs. So as you see, it is bigger, almost 3 times that E. coli bacterial genome size, but almost 10 times smaller than this number. So this number is not realistic. And as for this number, if we take a better look here, you would see that this is extremely small number, and if bacteria would have such extremely small probability to have a mistake per genome per generation, we have to choose the answer that correct number of mistakes per one bacteria per generation would be 0. So here we have one missing word, that is this number of mistakes per single base pair. So for example, here is messenger RNA. We have adenine, guanine, cytosine, and thymine here, and basically this mutation rate should refer single base pair, not the whole genome, but single base pair. So probability that this single base pair would have a mutation would be 1 out of this number going to have 7 zeros. So this is going to be 1, 1, 2, 3, 1, 2, 3, 1. So 1 out of 10 millions. And probability that this base pair also would have a mutation during one replication cycle also would be 1 out of 10 millions. And this one and this one. So for example, if we would be asked what is the probability that this base pair in the whole genome would mutate, we would say the probability 1 out of 10 millions. But what if the question would be, what is the probability that this fragment of the bacterial DNA would have a mutation? And this time our answer would be 4 out of 10 millions. So because we have to add 1 over 10 million plus 1 over 10 million plus 1 over 10 million plus 1 over 10 million, and the probability would be 4 out of 10 millions. So as you see, probability increased with the number of nucleotides. And if we would follow this logic, if we would have 10 million base pairs, we would have to divide 10 million by 10 million. So this gives us information that this bacteria would incorporate one mistake, one mutation per 10 million base pairs. And if we have 100 million base pairs, and if we divide this number by 10 million, the answer going to be 10. So let me clean the space, we don't need this number anymore. So if the size of the genome would be 100 million base pairs, if one mistake during replication would be 1 per 10 million base pairs, then we would have 10 mistakes in this bacteria. And this number, how big average gene, is absolutely not important for our calculations, and I will explain why. By the way, also you may find that these numbers are complete mess if you would divide total size of the genome. One more time, that is 100 million base pairs by average size of the gene, which is, according to our problem, is 2000 base pairs. We are going to get number of genes that this genome contains, and the answer would be 50,000 genes. And this is completely unrealistic number, because human genome, for example, is about 23,000 base pairs, and according to this problem, bacterial genome contains double number of genes in comparison with human genome. This is completely unrealistic. What is realistic numbers would be 5,000 genes. So this number would be more realistic, and that means that here we have a mistake. So instead of 10 in the power of 8, we should have here 10 in the power of 7. Then we will get 10 millions here, and if we divide by 2000, we are going to get rid of 1 0, and we are going to get realistic number of genes in bacterial genome. That is about 5,000 genes. And by the way, this number of base pairs per one gene is also not very realistic, because normal size of the bacterial gene about 1000 base pairs. But at least this number is close to real number, so we may leave this number. And also, as I will explain you now, the size, average size of the gene doesn't affect our calculations. For example, we may have 1000 base pairs here, we may have 10,000 base pairs here, and this wouldn't affect our calculations. So, according to our problem, we got 10 mutations per whole genome. And if the whole genome, even if we would take these numbers, would contain 50,000 genes, that means that 10 genes would have a mistake, and 49,990 wouldn't have a mistake. So once again, this is number of genes without mistakes, plus in 10 places we would have about 10 mutations, so we are going to get total number of genes 50,000. 10 of them would have a mistake, and the rest wouldn't have a mistake. So as you see, the correct answer would be answer C. But what if instead of 50,000 here we would have 5,000 genes? Would it affect our calculations? In this case, I would say that 4,990 genes wouldn't have a mistake, and 10 genes would have a mistake. So as you see, no matter what number is going to be here, and even if we reduce this number tenfold, still the probability that we would have, for example, 2 mutations in the same gene is extremely small. So, for example, if we would take this number, probability that 2 mutations would happen in the same gene would be 1 out of 5,000 multiplied by 1 out of 5,000. So the answer would be 1 out of 25 millions. So the probability that two mutations would happen in the same gene is extremely small. And if we would have increased number of genes, probability to have two mutations in the same gene would be 100 times smaller. But still, this number is extremely small, and new number would be also extremely small. So as you see, these numbers doesn't affect our calculations. So we can say that even with these numbers that are total mass, we would have about 10 mutations per genome. So we have to expect that 10 genes would incorporate these mutations. So we're done with these wrong calculations. Now let me switch to new slide where I will work with realistic numbers. This time my explanation would be much faster because I explained 90% of everything that I wanted to tell you today. So I change the problem, and subscriber who sent me this problem may approach his professor and ask for extra credit. So now this problem sounds how it should be, and all numbers are realistic. The error rate of bacterial polymerase is I reduced this number tenfold. Mistakes per, as you see, base pair per generation, and in our previous slide we missed this reference that per base pair. Before we should understand as per generation. That is completely wrong. So the size of bacterial genome is 10 raised 7, not 8. So this also would be realistic. Now we would have 10 million base pairs. That is normal. The size of average gene is 2000 base pairs. And actually the size of the average bacterial gene is about 1200 base pairs. But as I explained earlier, this number is absolutely is not important and doesn't affect our calculations. So how many genes are mutated per generation? I also have a comment about this number. Actual number of mistakes from the literature is 10 raised minus 8. So one mistake per 100 million base pairs. But as I said, realistic number of base pairs is 10 million per one bacteria. So what does it mean? It means that one bacteria per one generation during one replication cycle wouldn't have any mistakes, no mutations. Only 10 bacteria, when they would replicate, would have one mutation per 10 bacteria, or one bacteria per 10 replication cycles. So the answer should be 0. But let's say that this increased number of mutations would be due to some mutagens present, or for example that can be a chemical nature, or that can be for example some sort of radiation, for example ultraviolet radiation. So in this case, of course, we can see increased number of mistakes, increased number of mutations. But at least now this problem looks more realistic. So now my calculations would be very fast. We have to divide 10 millions, which is going to be the size of the genome of bacteria, which is normal size of bacteria between I would say 1.5 million and up to 13 millions, with average, like E. coli, about 4.6 million base pairs. And we have to divide by the probable rate of mistakes per one base pair, which is going to be 1 in a million. So we have to divide by 1 million. And as you see, we are going to get 10. As I explained at the beginning of my video, when we reduce this number tenfold, when we reduce this number also tenfold, we are going to get the same answer as before. And this is answer C. And as I explained earlier, number of base pairs per one gene is not important. One more time, if we divide 10 million base pairs of the bacterial genome by 2000 base pairs per one gene, we are going to get 5,000 genes. So now all these numbers looks realistic. And one more time, this number is like 100-fold bigger than normal mutation rate in bacteria. But we can get such a higher rate of mutation, as I said, if bacteria would grow in a mutagenic environment when harmful chemicals or radiation is present. That would increase, can increase tenfold or even hundredfold number of mistakes. And if number of mistakes would be even bigger, all bacteria would eventually die. And my last comment would be, as you see, if we would have 5,000 genes here or 50,000 genes here, 10 per 5,000 or 50,000 would be negligible small number, so we do not expect that these mistakes would happen in the same gene. And so we have to give an answer that we expect that 10 gene would incorporate mutations. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.